Okay. All right. How much time do I have left?
30 minutes.
40. Not quite 40. She's trying to cut me short.
You don't have to have 40.
It's just shy of 40. Okay. All right. Okay, well, we're going to look at another signaling pathway, maybe even two, but probably perhaps just this one. And this is a very critical pathway in development, in tissue maintenance and repair, and a whole bunch of other things. And when this pathway goes wrong, it is a very, very bad thing. Okay, and this pathway is known as the Ras-MAPK pathway. I know, you guys had so we went through this, but not everybody has had this. Okay.
Oh, Jessica was just really excited because she knew what you were talking about.
Oh, okay.
She's like, Ras.
Oh, yeah. Well, it is a very cool pathway because of its, shall we say, health implications. There's been a tremendous amount of research on this pathway because there we have a lot of targets for chemotherapy, for cancer treatment. Some of them are already out in the market too, by the way. But anyway, okay, this Ras-MAPK pathway is named after two of the major components. Ras, which is a G protein, and the MAPKs, which are a family of protein kinases. And this one essentially activates a chain reaction of protein kinases. You activate one small group of kinases, they phosphorylate and activate a second group of kinases, and then those guys go and phosphorylate and activate the third group of kinases, which gives you the final cell response. So we have a three-stage amplification. Not surprisingly, a little of the appropriate signaling molecules can go a long way because we're doing this three-stage amplification, giving a tremendous increase in cell response. Okay, now a couple things about this. It uses the monomeric single subunit G protein called Ras. And Ras is the granddaddy of the Ras superfamily of monomeric G proteins. Okay, so it uses that. But unlike the two pathways we've seen before, Ras never even touches the receptor. The receptor does not activate Ras directly. It activates some adapter proteins that then go and activate Ras. So we will say Ras is indirectly activated by the receptor. In other words, it does not bind the receptor at all. Okay, third thing, what does this thing do when we activate this pathway? Ultimately, it leads to phosphorylation of numerous proteins, including a whole batteries of different transcription factors. Oh, he's chasing me out. Oh,
Never.
Okay. All right. So when we activate the Ras-MAPK pathway, and that happens all the time in development and in tissue differentiation, tissue maintenance, wound repair, all kinds of things like that, what's going to happen is it can stimulate two major things, depending on the cell type. It can stimulate cell division because it can activate transcription factors that lead to productions of cyclins and cyclin-dependent kinases. It can phosphorylate things in the cell cycle directly, all kinds of fun stuff. So it stimulates cell division. Fix up that nice gaping wound you just gave yourself. Okay, and it stimulates cell differentiation. For instance, this pathway can get used in many cases. It's used as part of the thing for limb formation. If it wasn't for this pathway, you would have grossly malformed limbs because certain things activate this pathway in the stages of limb formation. And of course, when some of those things go wrong, then you get things like flipper limbs and stuff like that, no limbs at all. Think of thalidomide. By the way, thalidomide does not work by blocking this pathway. It has a different mechanism. but, you know, you can see the results. Okay, so it does a lot of things. Now, clearly, when you look at what it can do,
Is it used in like stem cells as well?
yes. Now with stem cells, in fact, it's widely, with stem cells, it turns out most of the time we don't know how to turn a stem cell into a particular cell type. It's been done for a few different cell types, and many times what it does involve, on the few that they actually know how to turn a stem cell into this fully differentiated cell type, you will use agents that stimulate this pathway. It's not the only thing, but it contributes quite a bit. Most of the time when they do stuff with stem cells, they take the stem cells, inject it into the tissue of interest, hoping that what's already there stimulates proper differentiation, and more importantly, this is a problem especially with embryonic stem cells, that they know when to quit. In other words, the cells know when to stop proliferating. Your job's done, stop. Because if they don't stop, what have you just done? You've given yourself a tumor. Yeah, and that has happened in animal and apparently a few human trials too. That's one of the biggest problems with embryonic stem cells, is telling them when to quit. We don't quite know how to do that all the time. So yeah, that's a risk of stem cell treatment. Yes?
Have you watched the South Park episode about stem cell research? I'm just curious.
No, I didn't. What was it about? What was it like?
Oh,
But-
you'll just have to watch it. I'm not going to talk about
Oh.
it. Very inappropriate, but I just wondered.
No, but I suppose that would be pretty interesting.
It's hilarious and very inappropriate.
Yeah, well, South Park's known for very inappropriate. They do have. You know, the funny thing, the other people who are great at satire is that what are often viewed as a kid magazine, that Mad magazine, which has been around since before I was a kid and stuff. Those guys really, their satire fights like a hungry shark, you know. They know how, they get you good. I've seen some great ones, and my God, you know. So it actually works at several different levels, but boy, when they go after something, they have very sharp barbs, and they stick them real deep into their targets. They got some really good ones. Okay, but at any rate, okay, back to this. Okay, we're stimulating cell division, cell differentiation. Needless to say, if this pathway is misactivated, that's bad. As a matter of fact, what we're going to see later on is a significant, any part of this pathway that's misregulated can push a cell to a cancerous state. There are several hot spots that do that. One of the kinase families is a special hot spot in certain kinds of cancers. Ras mutations in cells are found in about 1/3 of all human cancers. Receptor mutations that activate the pathway when you're not supposed to, those are another example. There are drugs out there now that are being clinically used that inhibit certain aspects of these pathways, and more coming in the pipeline. That's one reason why people want to know this pathway all the way down to the molecular level, because it gives more targets. Defects in this pathway are common in many types of cancers. So, you know, coming up with things that inhibit that pathway are going to be important. They're not cures, but they can certainly help slow things down a lot. And there are things out in the market right here and now that are being widely used for certain types of cancers. Okay, well anyway, we got this. Okay, now, how does the thing work? Well, before we map this pathway out, it's probably worth seeing what kinds of molecules activate this pathway. Okay.